Hi guys, so now we are on vision, and vision we're going to talk about photoreceptors. Okay, so let's start with what is light. Okay, so when you look at light, you can look at wavelengths for the light, and the wavelengths are going to correspond to different colors. One thing I wanted to point out is the wavelengths that our eyes are able to see is actually a very narrow range in comparison to all of the other wavelengths out there. Okay, so if you look at our very narrow range, 400 to 700 nanometers, and you look at the different colors, we're, basically think of like the rainbow, like Roy G. Biv. Okay, let's do some anatomy review for the eyeball. We're looking at a sagittal section through the eye. There are two general cavities. We have the posterior cavity that's going to be filled with a thick fluid called vitreous humor. And then we have the anterior cavity, which is in front of the lens. That's going to be filled with a different fluid that's all watery. That's aqueous humor right there. Okay, so anterior cavity, posterior cavity. And the eyeball itself has three layers. The outer layer is called the sclera in the back. Okay, that's the whites of your eyes. And then anteriorly, we have the cornea. Okay, so here's the cornea. That's going to be clear. Okay, so when you look at someone's eyes, you don't see the cornea because it's clear. And then the second layer here is going to be the choroid. That's called the vascular layer. And anteriorly, the choroid becomes the ciliary body, which contains the ciliary muscles and the ciliary zonules that hold the lens in place. And then in front of that, anterior to that, we have the iris, and the iris is going to control how much light enters the eye. And the hole in the iris is called the pupil. Okay, so note that the pupil is just a hole, a lack of structure. And then on the innermost layer, we have the retina. That's also known as the neural layer. That's where we're going to find our photoreceptors. And the last sensory neurons, which are called ganglion cells, they're going to come together to form the optic nerve, which takes sensory information to the brain. Okay, so when we look at the front of our eye, let's review. So the white part, that's the sclera. The cornea is going to cover the front of it. What we see is the colored part, that is the iris. The hole in the middle is the pupil. Note that it's just black because it's dark inside of the eye. Okay, so the iris controls how much light enters the eye. And the reason for that is if it's very dim, we're going to need to open it up. And so that's called dilation. And that's going to be mediated by the parasympathetic nervous system. It'll stimulate the muscles in there to dilate the pupil. And if we have too much light, we need to constrict the pupil. Also, constriction happens when you're looking at something close by to help get rid of the peripheral light rays to help focus the light better. But mostly it's to help, usually the iris is constricting to make sure that you don't have too much light so that you can protect your photoreceptors. That's going to be mediated through the sympathetic nervous system, and it's going to come in from the ocular motor nerve. Okay, so in lab, we went into the dark room and we shined the light into our eyes and we saw constriction. Do you remember the name of that reflex? That would be the pupillary reflex. Okay, so remember the ocular motor nerve, that would be the efferent pathway. Okay, let's talk about focusing light. Okay, so in order to understand focusing light, we need to talk about refraction. Refraction just means bending of the light rays as it passes through different media. Okay, so the amount that the light rays will be bent depends on the density of the medium that the light rays are traveling through. Okay, so for example, in this picture, we have the light coming off of the red straw. It's going to be passing through just air here. Here it's passing through the glass. That's going to, you see how it's a different angle here? That's because the light rays are going to be bent in a different angle because the glass is a different density. And then down here, the light rays are bent again differently because now they're traveling through water and then glass and then air. Okay, so each time it's going to be bent in a different, bent differently at a different angle, and that's called refraction. Now, when we're thinking about light entering through our eyes, think about the pathway that it has to take, right? So we're going to come from the cornea. We're going to travel through the aqueous humor, which is the fluid in the anterior eye. It's going to pass through the lens. Then it has to go through the vitreous humor. And then we're going to be able to get to the neural layer, the retina, where it'll activate the photoreceptors. Each time it passes through these different structures, because each structure has a different density, it's going to reflect or bend the light a little bit differently. So as the light goes through, it's going to be refracted each time. Now, we need to be able to control the angle of refraction in order to focus the light on the retina so that we have the best visual acuity, okay, make it look sharp. So of these structures, cornea, aqueous humor, lens, or vitreous humor, which one can we control? And the way that we're going to control it is by changing the shape. So the one that's going to change in order to focus the light is the lens. So that is the function of the lens. The lens is going to change shape in order to control the angle of refraction, to control how the light is focused on the retina. Okay, so let's talk about that. That's called accommodation. Accommodation is the changes that occur to the shape of the lens in order to focus the light when you're going from looking at something far away to close up, or from close to far away. Okay, so again, we're going to be changing the shape of the lens, and we do that using ciliary muscles, which are part of the ciliary body. Okay, so here's the cornea. This is the iris, there's the lens. So this round structure here, that is the ciliary muscles, and it creates a sphincter. Okay, so a circular, so that the muscle fibers, the smooth muscle fibers are oriented in a circle. And when they contract the diameter across will get, the diameter across that circle is going to shrink, right? Like constriction. So that means that the lens that is held onto by the cineral zonules, those are going to relax. Okay, so here's how it works. Okay, so we had a sphincter. When you are looking at something far away, okay, so if you're changing your lens to look at something further away, those ciliary muscles are going to relax. Now, because this is a sphincter, a circular orientation for those muscle fibers, that means that the diameter across is going to be greater, okay? It's going to go wider. And because it is greater, that means that the ciliary ligaments, okay, so the ligaments that hold the lens on, those suspensory ligaments, those are going to pull tightly on the lens. And if you're pulling the lens out in all directions, that's going to pull it so that the lens is flatter, or you could say less convex, but just means flatter. Okay, so when you're looking at something far away, the lens will be flatter because the ciliary muscles will be relaxed. Now, let's say that you're looking at something far away and then you look at something close up. Okay, so we need to change the angle of refraction by changing the shape of the lens. And we do that by contracting those ciliary muscles. Okay, so here's the ciliary muscles. They're organized in a circle. So when they contract, again, it's like a sphincter. It'll make it so that the diameter across is shorter. And if the diameter across is shorter, that means that those suspensory ligaments, also known as ciliary zonules, those are going to be looser, right? They're not held as tightly, they're not taut, okay? And so that means that they're not pulling on the lens as much, okay? And so because they're not pulling on the lens as much, that allows the lens to become rounder or more convex, okay? So far away, the ciliary muscles are relaxed, making it so that the lens is pulled tight and flat. Whereas if you're looking near, the ciliary muscles will be contracted, which means that the lens will be pulled, will not be pulled and allowed to kind of go back to a round shape. That's called accommodation. Okay, let's talk a little bit about vision problems. So if you have normal vision, right, everything's working properly, the light rays coming in, the lens is going to focus the light onto the back of the retina so that the light is all coming to a point at the fovea centralis. We'll talk about what that is in a second. Okay, and that's going to give you the clear, focused vision. Now, what happens if you have myopia, which is also known as nearsightedness? It means that the lights coming in, they're going to be bent, but they're going to come to a point or focus in front of the retina, which means that by the time they get to the retina, they're going to be scattered again. Okay, and that's going to result in fuzzy vision. Whereas if you have hyperopia, which is also known as farsightedness, it means that the lens is focusing the light behind the retina, so that at the point of the retina, the lights again, they're out of focus and they're going to be fuzzy. Okay, now astigmatism, that's a little bit more complicated because astigmatism can vary depending on the person. What that means is either the cornea or the lens itself is either asymmetrical, there's something a little off about it, or sometimes they have little extra bumps or grooves or something like that. And because of that asymmetry, it means that the light's not focusing properly and you're not going to get to it. That's why if you have an astigmatism, the doctor has to like really go and test every single thing because you have to have specialized glasses that will fix your particular asymmetry. Okay, now we get into the retina and the photoreceptors. Okay, so here we have the retina. The retina has three different layers, three different neural cells that we need to look at. Okay, so if we're looking at what's happening here, the light is coming in. Okay, so the light comes in this direction. So here would be, if we're looking at this picture, here would be like the inside of the eyeball, right, where the vitreous humor is. The light will travel past the ganglion cells, past the bipolar cells, and then it's going to activate the photoreceptors. Okay, so those photoreceptors, they're going to be up against where the choroid is. Okay, that'll then activate those rods or cones, and then the signal needs to travel back out. Okay, so the signal, the light comes in, right? Here's the inner cavity of the eye. The light bypasses both of those neural cells, activates the photoreceptors. Then the rods or the cones, they're going to activate what are called bipolar cells. They're called that because of their shape. They have two processes coming up of the cell membrane, the cell body. Okay, then we have the bipolar cells are then going to synapse with the ganglion cells, and then the axons of the ganglion cells will come together to form the optic nerve. Okay, so you do need to understand this pathway because we're going to talk about how the signal is going to go towards the brain. Okay, so those rods and cones, they pass the signal to the bipolar cells, which in turn pass it to the ganglion cells. Okay, let's look at the structure of the rods and cones real quick. Okay, they're named that based on what they look like, right? So we have rods that look like rods and cones that look like cones. It has to do with the shape of their outer segment. And in that outer segment, we have photopigment discs. Okay, so I'll show you what that looks like here. Okay, so here's a rod. There's its cell body. There's the shape of the rod. It's got stacks of these little pigment discs. And embedded in the membrane of those pigment discs, we have the pigment that's called rhodopsin. Okay, so rhodopsin is what it's called when we have retinal and opsin that are complexed together. And so what happens is when the rods are exposed to light, that rhodopsin is going to dissociate, which means come apart. And so it'll dissociate into retinal and opsin because of a chemical change where it changes the shape of the retinal, which then again makes it come off of the opsin. Okay, so the first thing that happens when you are exposed to light is what we call the bleaching reaction. And that just means that the rhodopsin dissociates to become the isolated retinal and the opsin. That's the bleaching reaction. Okay, now we need to, how do we take that and turn it into an electrical signal? In order to do that, we need to talk about what it does in the dark and then compare that to what it does in the light. So normally in the dark, the rods are going to be depolarizing and releasing a neurotransmitter, which is very counterintuitive. Okay, so in the dark, they release a neurotransmitter, but this neurotransmitter is an inhibitory neurotransmitter that makes it so that the bipolar cells are not, or hyperpolarized, are not activated, which don't send the signal to the ganglion cells. Now, in the light, the bleaching reaction is going to prevent depolarization, which prevents the neurotransmitter. And again, it's an inhibitory neurotransmitter. Okay, so it's like stopping. Okay, so it stops stopping, and what makes it so that when you lack the negative inhibitory neurotransmitter, the bipolar cells will then be stimulated to release their own neurotransmitter, which activates the ganglion cells, and then we're going to have signal to the brain. Okay, so let's look at that a little bit more closely. In the dark, we have what's called the dark current. And this is because when there is no light present, the sodium is going to be coming in, right? The sodium channels are open in the dark, which means we have sodium flooding into the cells. We call it the dark current because of the flow of sodium into the cells. Okay, so that's called the dark current because the sodium is coming into the cells, depolarizing the rods when it is dark. Now, when it depolarizes, that is going to cause the release of the inhibitory neurotransmitter. And again, inhibitory means it's going to stop, right? It stops the bipolar cells from releasing their own neurotransmitter, which means no signal. Okay, so dark current, sodium flooding in, depolarize to inhibit the release of an inhibitory neurotransmitter, which means that the bipolar cells are not activated and ganglion cells not activated, so we get nothing going to the brain. Now, in the light, the bleaching reaction, which just means the rhodopsin is dissociated, the dissociation of the rhodopsin is going to lead to closing of the sodium channels. When you close the sodium channels, you're closing the dark current. When you close the dark current, no sodium coming in, nothing positive coming in. That means that instead of depolarizing, we're going to hyperpolarize. Hyper meaning becoming more negative, further from the threshold. That means that we're going to stop releasing the neurotransmitter. Stop releasing the inhibitory neurotransmitter. Or stopping. It's like a double negative. Okay, so we stopping. Okay, so the no inhibitory neurotransmitter now allows the bipolar cells to become stimulated. The bipolar cells then release their own neurotransmitter to stimulate the ganglion cells. And now we've got action potentials going to the brain. Okay, so it's really strange. It's counterintuitive because of that inhibitory neurotransmitter. It's going to take some practice. Okay, let's talk a little bit about cones. Rods are good for sensitivity. Cones are good for color vision and visual acuity. Let's look at color vision. Now we have three different types of cones. We have one that responds to blue wavelengths, one that responds to green wavelengths, and one that responds to red. But we see a whole spectrum of colors. How do we see a whole spectrum of colors if we only have three cones, three different receptors? It has to do with the same thing we already saw with smell, a pattern of activation. Okay, so for example, if we have yellow light, here's where yellow light the wavelength yellow light, it is able to activate both green cones and also red cones. And that pattern of activation where you have green and red at the same time, that's going to be interpreted as yellow light. Okay, so again, a pattern of activation where you're activating more than one of the photoreceptors at the same time. That combination of the two, that's going to be coded for other different colors. Okay, so the other thing that cones are good for is visual acuity, which just means sharpness of the image and or resolving power. The way that you get a sharp image is by having more separate points. Okay, so for example, if you only got 4 things, we can barely see anything here. The more separate points of light that we're able to give us, the sharper the image or the more resolving power the image will be. Okay, so if you've ever shopped for TVs, I think you'll understand that, right? The more points of light we see, the sharper the image will be. Okay, so let's talk about how we get sharper images for cones than we do for rods. And it has to do with the idea of convergence. Convergence means coming together. Okay, so coming all together. So if you have rods, the reason that rods are much more sensitive than cones is because rods are going to synapse, or multiple rods will synapse with the same bipolar cells. Okay, so if you have multiple rods synapsing with the same bipolar cells, which will synapse with a single ganglion cell, so that if any of these rods are activated by light, you're still going to get a signal to the brain, and that's how you create sensitivity. However, when you create sensitivity using convergence, right, what you're losing is separate points of light. You're losing the resolution power. Okay, so if you have more, the more convergence you have, you're more sensitive, but you have less resolution. Okay, and the less convergence you have, the less sensitive you are, but you have more resolution. And this is why in the dark, you are able to see with your cones, sorry, wrong word. You're able to see with your rods, but you don't see very well. It's not very sharp. Okay, so let's compare that. So for rods, again, we're very sensitive because we have multiple rods coming together to activate bipolar cells, and then that activates, a ganglion cell. Whereas cones, we're not going to have any convergence, especially in the fovea centralis. Okay, so if you're looking at the retina here is the optic nerve. Above that, we have the fovea centralis, which is a region of the retina that is populated just with cones. So if you're trying to focus the light and be able to see clearly, you want that light to focus on that fovea centralis where the cones are found. And the cones are going to be good for visual acuity because they do not converge. You have one cone going to one bipolar neuron going to one ganglion cell. Okay, that means that your brain is going to receive signals from every single one of these cones individually. We get more points of light, which we see as a higher resolution. Okay, to sum that all up, rods are more sensitive because they have more convergence. Cones are less sensitive, but they have higher resolution because they have less convergence. Okay, so which photoreceptors allow you to make your way to the bathroom at night? Okay, so we're looking for light sensitivity, so that would be rods. Reading small print, we're looking for visual acuity, so that would be cones. Enjoy the colors, which ones have color? Rods are just black and white, so that's going to be cones as well.